We are pleased to have Dr. J. Craig Venter as our guest throughout the hour, the leader of the team that created the first synthetic cells. We're also back with science author and writer at The New Yorker, Michael Spector. And joining us now is bioethicist and co-founder of the Columbia University Center for Bioethics, Dr. Robert Klitzman, and author and biological and chemical weapons expert, Jonathan B. Tucker. We have just heard about the amazing potential of Dr. Venter's breakthrough, but along with this potential for good, there is also potential for evil. We're going to take a look at a clip now from the documentary.
There's clearly people in the world that like doing harm to other people, and this is, in theory, giving them one more way to do that. There ensued an incredible government review, including several sessions, I've been told, in the White House, trying to decide whether to make our work military secrets or to have us publish it, what to do.
Do you want everyone in the world to know how to do it? Do you want every high school kid to know how to do it? Do you want terrorists to know how to do it?
This is actually debated in the scientific community as to whether or not anybody could actually design an organism that's more dangerous than the ones that exist naturally. And
that is a pretty scary jumping off point, Jonathan, for us to talk about bioterrorists and if they could actually use this technology to make more dangerous weapons than exist today.
I think in the distant future, but there are other near-term threats that I think are a much greater concern. I think at this point, the technology is cutting edge, way beyond the capacity of terrorist organizations. So it would really be in the hands of states to misuse this technology effectively. And what they could do in the near term is simply recreate dangerous pathogens that are difficult to acquire because they are very rare or have been eradicated or are very limited. But this technology is moving so quickly that I think within 10, 15 years, the technology will be democratized. There will be kits that enable graduate students, college students, perhaps even high school students to begin to assemble bits of DNA called biobricks with defined function to create genetic circuits and even entire artificial organisms.
That's what terrifies me, frankly, because you do read these stories that if you're able to take your computer codes and you get the right chemicals and you buy some equipment off eBay, that you could do this in your garage, as you just suggested.
That thrills me. I think because if it's one thing we know about humanity, we are already capable of doing immense harm to each other. We don't need special new tools to do it. It's not like suddenly Craig has invented something that'll let people kill other people. What we don't ever talk about are risks and benefits. And you need to think about the risks. They're very serious, they're real. But there are also benefits, and we need to talk about both of them. And most people either focus on the risks or they focus on the benefits.
What is this balance that you think needs to be struck? And on one hand, honoring what you were saying, Michael, the great promise of this technology, and at the same time also respecting what a lot of people in the American public are saying is this is downright scary.
This is a discussion that's been going on for at least 30 years that I've taken part in it. My team, when I was at NIH, was actually asked to sequence the smallpox virus on behalf of the U.S. government, because the theory was after it was sequenced in the U.S. and in Russia, the remaining stores of the virus would be destroyed. Upon sequencing the virus, we found out how similar it was to other pox strains, including what we're immunized with, or what we've all been immunized with, and that was vaccinia. And I argued back then there was no point in destroying the virus because it could be recreated even with some minor changes or a new evolution from monkeypox viruses and other things. So it would have created a false sense of security when in fact there was none.
People are not only concerned about the bioterrorism risk we've just talked about. There's a great deal of fear about potential accidents in the lab. There's also a lot of fear about unintended consequences, things that could happen that we can't even imagine today.
The real challenge with synthetic biology is that it requires a new type of risk assessment. With recombinant DNA technology, you're taking a gene from one organism, one or two genes, and transferring it into an organism that is well characterized, well understood. The idea with synthetic biology is you have Lego blocks, pieces of DNA that can be assembled into circuits and modules that provide new types of functioning that do not exist in nature. And there is a potential for interaction among these genetic elements that is unpredictable, what's called emerging properties that cannot be predicted on the basis of the parts themselves. And so how does one calculate the risk of an accident or unexpected pathogenicity? Because this has never been done before. It's unprecedented.
It's interesting you should say that, because I have a question from a Facebook user who poses almost the identical question. He says, what if this so-called synthetic life mutates and spreads out of control in a deadly fashion?
I think these are risks. I mean, I agree that we need to balance the risks against benefits. It's possible for dangerous viruses to leave a lab, no matter what protections we have, and to affect human beings. Is that extremely likely? No. Is it possible? Yes. I think that the actual where we decide to regulate or what we decide to do gets quite hard and complicated. So I think that we need to think how to deal with these issues.
Let me put this in context.
Put it in context, because I want to know, just specifically, how concerned are you personally about the kind of accident where someone is working in a lab and a gene mutates and
these are the same arguments that have been going on for decades and decades since there were first tools in molecular biology. And there's probably been tens of millions of experiments done in research labs around the world where genes from every known species have been put into the bacteria E. coli without a single incident or a single problem. Why? Because the genome was changed so that bacteria could not survive outside of the laboratory. That's one of the guidelines that we're trying to build in and get the field to adopt with synthetic genomics. Of any new organisms that built cannot survive outside of the laboratory. So there's been nothing that's ever been engineered in the lab that's on its own mutated to get out of control.
What do you say to the critics of this technology out there who feel that you should have a moratorium in place until you can fully explain the risks involved?
This is what I
too
say.
far.
If you did that, that's called the precautionary principle, and it basically says we shouldn't move forward with something unless we're so certain that the risks are non-existent, that it's safe. And if we did that, and we would not have airplanes or x-rays or antibiotics or vaccines or any modern thing that you name, because there's risk entailed in every single thing we do.
Right. I think moratorium is too far. I think, in my mind, there needs to be ongoing discussion. In the end, how do we weigh the benefits against the risk is really a political decision. It's a social decision. For us as a society to decide, we want science to go forward. We want the benefits of that. We're also aware of the potential for terrorism or things going bad or accidents happening.
I think there are different types of threats. Toxic chemicals do not reproduce, so we can manage that risk in a much more predictable way than with a new type of self-replicating organism that once it got out from the lab could interact in unpredictable ways, both with human health and with the environment. So I think it requires a greater degree of precaution than we would with other types of threats that we deal with in everyday life. In 2001, a group of Australian scientists who were developing a vaccine to control mice populations were experimenting with mousepox, which is a benign, relatively benign virus in mice. They transplanted one gene into that virus in order to enhance the immune response to make the vaccine more effective. And unexpectedly, they made that virus into a killer virus that killed all of the mice in the experiment. In retrospect, they could understand what had happened. It had knocked out the mice's immune system. But that was an example where a single gene had an unpredictable, very lethal effect.
It's essential to mention the new NIH guidelines that have been proposed that require all the companies that synthesize these DNA fragments to have to screen them against the A list of infectious agents so that somebody couldn't frivolously try and make something that's harmful. It doesn't block them if they get their own DNA synthesizer, but it stops any frivolous uses where somebody can just order something. So I think the first line is already being proposed of guidelines. We argue they actually didn't go far enough.